In this episode, we'll explore what makes stem cell therapy safe. My guest is Dr. Carlos Escobar, a medical doctor with a master's degree in molecular biology and biotechnology and a PhD in biomedical sciences. Carlos's PhD focused on improving the safety and effectiveness of stem cells. Carlos's lab has supplied clinical-grade stem cells for more than 10,000 patients. In this conversation, he explains why certain types of stem cells should never be used, why many labs are no longer using bovine serum, and why contamination is one of the biggest hidden risks in regenerative medicine. At the end of the video, I'll cover the critical questions that you should ask before choosing a doctor or clinic. We begin this conversation by discussing why not all stem cells are created equal and which types are safe to use and which types aren't. Before we get started, I recommend that you turn on the subtitles that I've created for this episode to help you get the most out of the conversation.
First, I think we need to clarify something. There are different kind of stem cells, right? Stem cells, it's a very broad term. So you could have, for example, totipotency stem cells. You could have pluripotency stem cells, that these are the embryonic stem cells.
Yeah.
And these two are not safe to use on treatments, right? So first, I will need to clarify that you have to use adult stem cells, adult on the biological standpoint of view. So these will be cells that you extract from tissue that have more than 9 weeks of gestation. I will say that every single tissue that you extract after the birth of the baby, you can extract adult stem cells. You can use any different kind of stem cells, but always should be extracted from tissue after the birth.
Okay, because
Right.
that makes it safer.
Exactly. Because before is not safe. There are a lot of reports showing that the cells that you extract from tissue before the birth, you can have a cancer after that.
Okay.
But with the adult stem cells, it's perfectly safe.
Got it. So the first thing is you need to get stem cells that are adult to make it
Adult
safer.
on the biological standpoint of view.
Mesenchymal stem cells are the most commonly used stem cells in regenerative medicine because they support healing and have a very low risk of immune rejection. In the next section, Carlos explains where these cells come from and why donated umbilical cords are his lab's preferred choice, and why proper donor screening is so important. In the process of making stem cells, you use umbilical cord stem cells?
So you can extract mesenchymal stem cells from different tissue. I mean, skin, right? Fat, the teeth, right? The bone marrow, right? The umbilical cord. So why do we use the umbilical cord? Because it's a very young tissue, right? And you can characterize the person who donate this tissue. Be sure that, for example, this family doesn't have cancer, to trying to avoid cancer challenges on the cell, but also these people doesn't have viral infection, doesn't have drug consumption, doesn't have a sexual violence story, right? You can perfectly characterize the donor.
Okay, and that's important because
Totally important.
the cells that you extract, if the person was sick and had maybe a virus or had some
Exactly.
disease, that could pass through those cells to
Exactly.
the person receiving them.
Exactly.
In the next section, we explore a critical issue that is often overlooked by many patients: the growth medium that is used to expand stem cells, and specifically why many labs are no longer using bovine serum, which was standard up until recently.
I mean, this industry has a huge problem because everybody is using a solution extract from calf serum.
calves, from
calves.
cows,
Exactly, but fetus.
the fetus of the cow. And if I understand, using animal products to make human stem cells, to grow them as like a growth medium, has some risks.
A lot of risk. A lot of risk. Think about this. For example, around 50% of people has antibodies against molecules from cows. So if you produce cells with molecules from cows, you will have at least 50% of the people rejecting the cells.
Wow.
But also, using growth factors from cows to induce human cells growing is not a perfect fit. So the cells grow very slowly and get old pretty fast. So at the end, you have not the best product to treat your family, not the best alternative because of the rejection, but also this product from the cows is so expensive. So at the end, you have a bad product, expensive, and unsafe.
And your lab has produced stem cells in a novel way, using a different source of production that makes it safer, or can you just
Eh,
share about that?
I develop a technique to extract growth factors from people blood. So I receive um, plasma samples from blood banks in Colombia, and I organize and mix it and I extract a couple of proteins, and using that we induce cell growing on stem cells. And using this, we avoid the problem. But also, this take us to produce the cells in a very cheaper way. But also, the cells grow amazing, um, pretty fast. But also, the cells are pretty healthy. So we found an excellent way to produce the cells. So based on that, the university set up the lab, and I modify the process to produce the cells based on those results.
Wow. Let's talk now about the risks of stem cells when you're making them. What could go wrong?
So my short answer is, if you have an adult stem cell well produced, it's safe. The cells itself is safe. But to guarantee the cell surviving and growing, you need to guarantee good biological conditions. And those conditions will support growing and surviving of microbes also.
So what you're saying is that as you take a cell out of an umbilical cord and you start to expand it or to grow it, then the exact same conditions that make those cells want to grow also make other things that you don't want in there
Exactly.
grow as well, like bacteria
Exactly.
or molds.
So you have to prepare everything looking to avoid presence of any other kind of microbes.
And how do you do that?
A lot of work. It's a lot of work. It's a lot of work because it's pretty interesting. For example, I know that there are several labs that use antibiotics on the cell production process, and also use antifungal. So we understand that if you put on a cell culture system antibiotics or antifungal, those molecules also will damage the cells.
Okay.
So we don't have it, right? So I say to my team, you have to think that you work with something like an intensive care unit. So you have patients on your intensive care unit. So you have to review, you have to see it, you have to visit them almost every 2 hours. So we have people just being cared that every single cell is in a perfect condition. And if we see something bad, we have to discard.
Got it. Okay. So You are making the cells in a lab, you're growing them, and then avoiding that they get contaminated.
Exactly.
Then what happens?
We have a system when we can tell how is the health of the cells, right? So we take it usually until third or fourth passage, and after we recover all the cells and freeze it in very tiny vials of glass, right? So we have very tiny vials sealed, and we need to be sure that we don't have any kind of microbe on those vials. So let's suppose that we have 200 vials and we seal it and we freeze it. So tomorrow, somebody not from the production team choose 10% of the vials, and we send it to another lab, a third-party laboratory, and they test to guarantee that we don't have any kind of microbe on those vials. So because we have everything sealed on the same time with the same person, with the same team equipment, we can infer that if we don't have something bad on those vials we send to this lab, we have nothing on the vials we have on the same batch,
Got it.
right? So we test after all the manipulation, all the production process, we test everything. And if everything is on place, we allowed using these vials to run treatments.
So that's the end of the process. So the
Yeah.
testing on the final product
Uh-huh.
to make sure that the same product that the person is getting, like
Uh-huh.
that has been tested to be safe.
Exactly. Exactly.
And what could go wrong if, for example, there was a little bit of mold or some bacteria and it got into a what could happen?
It's a nightmare to think that you have a bacteria growing in the body of somebody, right? But I mean, I have heard some labs that had a couple of patients with infection in the brain because of the cells that they put on the blood. Think about this: it's an infection on the brain after putting the cells on the blood. So you can have an infection any place. So for sure, the minor consequence should be that you will be very sick for a long time. But these cases, I'm telling you, everybody have died. So
It's a big deal.
no, for sure, for sure.
Now, there are labs making stem cells from the doctor who is kind of just taking out some stem cells using a syringe in their lab, and they're like just injecting them again. So there's no kind of expanding. Then there are people who are a little bit more technological. So they maybe have a little lab and they're making some stem cells in their clinic, right up to more sophisticated procedures. It sounds like it's a little bit risky.
A little. It's impossible to be sure that you don't have a bug, you don't have a microbe. Excuse me. Bug is a bad term. But it's impossible to be sure that you don't have a microbe if you don't have a proper test. And you can't have a proper test before 4 or 5 days. It's impossible
You mean it takes that long to test?
to be sure that
the
you don't
cells?
have it.
So what you're saying is,
If you prepare something at our lab, if you prepare something, yeah, we have everything. We have the clothes, we have the people trained, we have everything, the lab, the filter system, everything. But anything can happen, and if something goes wrong, and you put something on somebody body with a bug, I mean, doesn't make sense. If I mean, it's at the end, you need to be sure that you will help people, not hurt people, right? You need to be sure that you will help them. So so to be sure that you don't have a bacteria, you don't have a fungus, is a big deal. It's pretty important. So it's impossible if you prepare something and you put in somebody,
Straight away.
yeah,
You just mix it up and put it in.
it's impossible to be sure. And there is no way.
Got it. Okay, so let me see if I'm understanding. So the only way to know that the cells are guaranteed to be safe and not have any contamination in them is to test them.
For sure.
And to test them takes maybe 5 days. And if you don't freeze them while they're being tested, then
They will die.
They will die. So your solution is to freeze the sample. That gives you enough time to test
freeze
them.
everything. Freeze everything. And I choose randomly some vials and send it. So if this sample choose randomly from the batch is clean, I can infer that all the batch is clean.
Got it. And this is so important because stem cell therapy is still not completely regulated. So people are putting in stem cells all the way around, and not necessarily we're learning about the problems that are happening because it's not regulated. So it's a big issue of safety and making sure you're getting them from a safe source.
Yes, yes. But also I will say that, I mean, this is happening. I will say that this will change for good the medical exercise, the medical work. So we need to be sure that we are doing properly everything.
I hope this episode has been valuable. Before you go, I want to leave you with a short list of questions that you can ask a physician or clinic offering stem cell procedures. Asking these questions isn't being disrespectful or difficult. It's simple due diligence, and it helps to ensure that the people you're entrusting with your health are actually prioritizing your safety and treatment outcomes over convenience or cost. Here are the questions. First, where do the stem cells that you propose to use come from? Are they going to be my own cells or are they donated cells? And if they're donated, how are the donors screened for infections, diseases, or cancer risk? If the cells are my own, how do you assess whether my stem cells are present in sufficient quality and quantity to treat my condition? This is especially important as we age, as the number and function of our stem cells decline over time. If the stem cells are donor cells expanded in a laboratory, what is the growth medium used to expand the cells? Are animal products like bovine serum used? Finally, how is the stem cell product tested for contamination? Is testing bacteria, fungi, and microbes performed by an independent third-party lab? And can you review the test results for the specific batch of cells that would be used? I've put together a downloadable PDF with these and additional questions that you can use when evaluating a stem cell clinic or physician. You can find it at yourstemcellguide.com/PDF. And there's also a direct link in the description of this episode. Thanks for listening.